Processes in Recombinant DNA Technology Part 3. This is continuation of the first two discussions. In this, I'll start from the sixth step, that is insertion of recombinant DNA inside host. I repeat, I'll start from insertion of recombinant DNA inside host. First 5 steps are discussed in earlier part 1 and part 2. Now coming to insertion of recombinant DNA inside host. Sixth step, insertion of recombinant DNA inside host. Insertion of recombinant DNA inside the host to insert the foreign DNA or the vector. Recombinant DNA means the vector DNA carries the desirable DNA fragment, that is recombinant DNA. This recombinant DNA is inserted or it is put inside the host cell, the carrier cell, which carries the recombinant DNA, and along with the host, the vector DNA also replicates and produce a desirable product in large scale. Basically, bacterial cells are used as hosts because their copy time, that is time required for replication, is very less. Now coming to insertion. To insert the cell, first the host cells must be made competent, that I discussed earlier, preparation of competent cell. Now coming to how to isolate or how to distinguish between the host carrying the recombinant DNA and the cells which do not carry recombinant DNA. I repeat, how to distinguish between the cells or the host cells carry recombinant DNA and the cells which do not carry recombinant DNA. How to distinguish or how to understand. For this, selectable markers are used. Selectable markers are used to distinguish between the host cells carrying rDNA, that is recombinant DNA, and cells which don't carry rDNA. Now, how it is done? What is selectable marker? Selectable marker is a specific property, and it is used means the separation or distinguishment is done by either the presence or absence of selectable marker. For example, antibiotic resistance gene, ampicillin R. Suppose for example, suppose one rDNA carries ampicillin resistance gene. As a result, recombinant cells means the cells carrying the host cells carrying recombinant DNA, that is host cell carrying recombinant DNA. So as a result, recombinant cells will grow in a medium containing ampicillin, whereas non-transformants, that is non-transformed cells, and I've explained what is transformation also earlier in the earlier recordings, whereas non-transformant will not. So this is how selectable marker is used to isolate or distinguish recombinant cells from non-recombinant cells. Now coming to next part, that is large scale production. Large scale production, next topic. Large scale production. So after we introduce or insert the recombinant DNA inside the host cell also reproduces. Next point, that is large scale production. Once vector DNA or recombinant DNA is inserted into the host, that is host contain recombinant DNA, then it is used for large scale production. Now what is large scale what is used for large scale production? The container used for large scale production is called bioreactor. As you can see, it's written here. So a large scale production is done in bioreactor, and either continuous culture system or batch culture can be used. Continuous culture is preferred because it is much more, the product yield is more. So for large scale production, what all we must know, that for large scale production of the desirable product can be the gene itself, or just a moment, continuous culture is used as medium. Okay, as I was saying, that the desirable product may be the gene itself, or the protein, or any other substance obtained or produced or coded by that desirable DNA fragment. Now why continuous culture is preferred? Because continuously and what is continuous culture? It is a type of medium in which continuously fresh medium is added, and the waste product as well as the end product is extracted or excreted. As you can see here, the continuous culture is where the used medium is drained out from one side, while fresh medium is added from another side to maintain the cells in their most active state, physiologically most active state. That's why continuous cultures are preferred. Active stage, that is called exponential phase of growth. So why continuous culture is preferred? I repeat, because what is continuous culture first? just a moment. Continuous culture is a type of medium in which fresh medium is added from one side and discarded. Used medium is discarded or removed from another side. Now, why continuous culture is used? Because it is continuous culture. This culture is used because it helps the cells to be in most active state or in exponential phase of growth. Now, what is a bioreactor? A bioreactor is a container in which raw materials, individual enzymes is obtained. So, what is a bioreactor? Bioreactor is a container in which large scale production is done, in which raw materials, individual enzymes, etc. are added, and optimal conditions for growth of the specific organism is maintained. Now, what is optimum growth conditions? Maintenance of pH, temperature, substrate, salt, vitamin, oxygen concentration. I repeat, bioreactor is a container you can say it is a vessel in which raw materials are biologically converted into specific product, individual enzymes, etc. using microbial plant, animals or human cells, and it provides optimal growth conditions for achieving desired product by providing optimum growth conditions. And optimum growth condition means maintaining all these factors so that the growth is maximum. Now, most commonly used bioreactor are stirring type, so that here stirred tank bioreactor, so that sufficient amount of air can be added, as you can see in the image. And now coming to the stirred tank reactor. It is cylindrical with a curved base to facilitate mixing of reactor contents, and the stirrer, as I told, facilitate mixing of the components as well as increase oxygen availability. And this type of bioreactor consists of an agitator system, an oxygen delivery system, a foam control system, a temperature control system, pH control system, and sampling port. Sampling port collects small volume of culture periodically. Now, what is downstream processing? That's the final step. As you can see, the last step, extraction of desirable product, is done by downstream processing. Now, what is downstream processing? It is the processes which include separation, purification of the desirable product, and then quality control of the product is done. So, downstream processing, last point you can say that extraction of desirable product is done by downstream processing. And what is downstream processing? Extraction and purification processes or steps of a desirable product is collectively called downstream processing.